Hey there scientists, and welcome to a video demonstration for Science Rendezvous. Now, if you've done any of the DNA extraction videos, you might have noticed that DNA kind of looks like these white, thin, little thread-like thingies, like it just doesn't seem there's much to it. You're just kind of hanging out in water, they look white. I mean, it's cool to see them, but it doesn't really seem there's all that much to them. Like, what's the hype? But when people talk about DNA, they say something like this. They say, oh, DNA is the language of your body. It's like these letters that are arranged in certain sequences and certain patterns, and your body reads them like words and says, hey, if my DNA says this, therefore my hair color is black. And really it's made up of these 4 building blocks of G, C, T, and A, but when you look at these white threads again, you're like, I don't really see them. Where are they? Well, the idea is that in order to go from those white threads to this language and these words, you need something a little bit more. And turns out there's this field called bioinformatics. Now, this might seem like a really long, scary word, but all it is, it's the application of different fields, such as, and especially, computer science, to biology in order to answer the question we just asked. How do we see these letters on our threads? And what they've done here is that they've made these really nifty tools in order to visualize DNA and do a lot more with them. And what's going to happen in this video is that I'm going to introduce you to one such tool.
So here we are. This is the nice, neat startup page for Eugene. Now, I said that this tool allows you to see the 4 blocks of A, C, T, and G and the patterns they make on your white threads. So let's take a look. Now, right now, you can't really see it, right? You can't really see the A, C, T's, and G's. All we see are these little blue and yellow arrows. Now, these arrows aren't meaningless. They actually tell you that there's something special about the pattern in this area. So how about we zoom into one of these patterns, right? And see what's up. So let's select this area right here. Let's tell Eugene to zoom into this area right here. And so, okay, now it's getting a little bit, but we still can't see our A's and C's and T's. So as we zoom in more and more, we start seeing them. So what this is telling us actually is that this pattern right here, this pattern that A, C, T, and G make actually has a special name, and that's this name right here. Now, I know this name has a lot of numbers and it sounds really boring, and that's an unfortunate fact about science, that sometimes, the names are kind of boring. But hey, this is a little bit of a cost that you have to pay for doing amazing science. Sometimes you have to have boring names. But this is great, right? So now we know that, hey, this has a special name. Maybe it has some special features. Maybe I could compare it to other species, and maybe I compare it to other organisms. So now I'm thinking, we're all busy scientists, right? Like, we do a lot of different DNA extractions. We're not just out here, extracting banana DNA only. We might also be, doing DNA extraction for peaches and strawberries and oranges and who knows, maybe different plants, even. So we have all these different patterns. We have all these different sequences, and we're thinking, is there a way that I could compare these different sequences and different patterns all together at the same time? Will Eugene let me do that? And turns out, it actually does. Now, here I don't have the sequences for banana and strawberry and, different fruits, but what I hope you can kind of see here is that they are all still very different. So now you can see all these different sequences. So for example, the first one right here belongs to this one. This sequence belongs to this. As I click through, I can tell exactly which sequence belongs to which organism. Not only that, Eugene has automatically compared all of the sequences together. It has compared all these patterns and given all these different colors to make it even easier for me to look at this. G is in blue, C is in green, T is in red, and A is in yellow. And because of these different colors, I can already start to pick out that, hey, between 22 and 23 or 22 and 24 right over here, all of these sequences have A's in them. Huh, is that something interesting? What's going on over here? Or maybe you've seen even a bigger block. For example, right here, you might notice that these three sequences actually share a lot of different sequences together. All of these sequences share, A's, T, C, G, A's, G, C, T. Like, so many of them have this right over here, except for this one. But now I'm thinking, is it possible that the ones that are more similar are somehow more related to one another? Now, many of you have done what's called a family tree in perhaps class, where you have yourself, and then you go to your parents, and then you go to your grandparents, and your great-grandparents, and you try to go as far back as you possibly can in your ancestors. And now what I'm telling you about, what the kind of tree that I'm talking to you about, is actually not all that different. I just want to see how related these are. And, are there any grandparents? Can we see them? What's the relationship? And so what Eugene allows you to do is to build a tree. So if you see over here, there's this really cute little icon of a tree right here. And if I tell it to build a tree, it will build it for me. So now let's get rid of some of the extra stuff on the screen so we can see here. These sequences are really pretty, but we're not looking at them right now. We just want the names of the organisms, and we want to look at our tree. Now, if you look at that. So now here I want to highlight something. As I move across this tree, it's telling me how related each of these are. So now, because I'm starting at the bottommost level or at the rightmost level, all these different species have a different color because, they are all fundamentally different. But as I move this blue line across the tree, move it further up, I start seeing that these different species start changing their colors. And the ones that share the same color are probably in the same group. They might be sisters or brothers or something like that. So maybe this bicolorana, bicolor, and rosellana and roselli are actually a lot more related to one another than they are to this montana. As I move further up, I can start seeing even bigger groups. I move even more up, I see even bigger groups. So it's telling me that these four, that even though bicolorana and rosellana are more closely related to each other than montana, but all these four together are actually a lot more closer to each other than either of them are to this Gamsoclias. And as I move up, again, I get bigger groups, even bigger groups, until all of them pretty much have the same color. And so now you can tell that the ones that don't share the same color are probably the most different in this entire group. So now that's great, right? And now we can get to see these different colors, you get to see, all these different patterns, where they match, where they don't match, how related each of these different organisms are. But let me tell you a little biology, let me tell you about a little bit of science. So far we've talked about DNA, right? But DNA doesn't actually act alone. DNA is more like that bossy teacher that tells you what to do, right? DNA is kind of like, hey, this is what I'm telling you to do, these are the codes, these are the instructions, and this is what I want you to do. But the thing that actually carries out the work, that actually carries out those instructions, that does the work, is what we call proteins. Proteins are actually the things that give you your eye color and give you your hair color. In fact, your hair and your nails are made up of proteins called keratins. But enough of that. So far we've looked at DNA, but can we, instead of looking at, teacher, can we look at the thing that actually is doing the work? Can we look at these proteins? And actually, Eugene lets you do this. So let's take a look over here. So this is, as I haven't said this to you yet, but proteins are essentially blobs that kind of are in your body that just kind of hanging out. They're not just hanging out, they're doing some really important work. But why I call them a blob is because they have this really funky looking, structure. They're kind of just like a bunch of spirals and arrows and different rods and different shapes. But these are actually very important. And what's interesting about proteins is that so far we talked about the pattern in DNA, right? Like DNA has these 4 blocks, the C, G, T, and A, just 4 blocks, that's it. That's what makes the pattern, different arrangements, that's it. But for proteins, you actually go a step higher, a step more complex, because now you don't only just have these 4 blocks, you also have 20 blocks. So 20 different blocks arranged in different things. You can just think about the different arrangements and think of the different possibilities for the different proteins that we have. And what's interesting here now is that we can see this protein in Eugene. And maybe I'm interested that, okay, I'm looking at this region right here. What if I were to just select this region? Can I see where exactly it is? And look at that. As I move across, as I select more and more of these blocks in my protein pattern, I can see Eugene highlighting it on the protein structure. Let's see right here. You can see the yellow right there. There's the yellow right there. And maybe I can keep going, right? So if I keep following the yellow, see where it's ending up. So what this is telling me is that this pattern right over here corresponds to this yellow structure right here in my protein structure. So that's pretty cool, right? Because now I can look at this structure specifically and see, hey, what's cool about it? How is it different from other regions? What else can I do with it? Is there something that's interacting with it? What is its special features? All these different questions suddenly become possible because I can look specifically at this region, and it's super easy to do that in Eugene. But I think for now,
that's it. I hope you learned something new from this video, and thanks for tuning in, scientists. I'll let you get back to your experiments. Bye!